Hi, everybody. We'll just give everybody another minute to join there. I hope all's well. You might just let me know, as I always do. You might just let me know if you can hear me okay. You can see the notes on the screen. Give me a little thumbs up or a quick hello. And we'll get started in another minute or so. We won't start just yet. The notes for those that are attending this week free. The notes for today, hi Sean, are on the news feed. It's I put up 2 handouts: cell structure, cell diversity, osmosis diffusion. How are you, Clodagh? How are you, Caitlin? Shannon? Happy Thursday, Cal. Good. Hi, Anna. All right. So we'll just give everybody another second there. So, and as I say, we have another big class. This is the final one of our biology blitz this week. But as I said, we're going to have a lot more in the next couple of weeks as well. All right, so we'll just give it. Just gone 31 minutes past. Okay, everybody, we're going to get started. So. On average, hello everybody, good to see everybody, thanks for all the hellos. On average, I think there was about 340 people watching the class yesterday, and about 290 or 300 the day before. So it's been in around 300 or a little bit more than that over the last 4 days. It's been brilliant, absolutely amazing. And just the feedback that a lot of people are saying they found it so productive. So that's why we're going to do a lot more of these up in the next 4 or 5 weeks. Let me just remind you. This is what we've done so far. All right. So let me just get my pen here. So look. This is what we've been focusing on. So we've done cell division, enzymes, photosynthesis, respiration. Now today, we're going to do cell. I'll put it in blue here. Today we're going to do cell diversity, cell structure, and we're going to do osmosis diffusion. So we're going to do these 3 chapters today, and then that's it. And then hopefully, then we can say to ourselves that by doing these 7 chapters and knowing these 7 chapters really well this week, we'll have 30 to 40% of our leaving cert. Last year was actually a little bit more than that. And remember, it's all about the question bank as well. Those questions you must be able to do within those chapters. Now, because this is our last class this week for our biology blitz. So I know not everybody is here. Not everybody is a paid member, so therefore, obviously, this is just for people who are members all the time. So look, this Sunday, we're doing variation and evolution, and we're doing genetic engineering. So that's it. The last couple of Sundays, we did DNA and RNA before that, and then before Easter, we did 2 2-hour classes, 4 hours in genetic crosses. So that will finish up the genetic section. So that means then we'll have all of the genetic section done over the last 3 or 4 Sundays. And then on Tuesday, I've never done the lymphatic system with you, so I'm going to do that again, or for the first time. But I've done the senses already about 3 months ago. We're going to do the senses again. And then on Thursday, we're going to do and finish off plant biology, plant reproduction. There's a little bit more to do on that. So that's the plan for next week. Now, let me just clarify this because I want to make sure everybody knows the plan for May. This is what's going on in May at the minute. So look, you know my H1 biology feed, my platform there. You go to the very top. There's your notes. There's your plant biology notes. All your other notes are in that digital book. There's your question bank and your solutions to your question bank. And finally, the schedule. So look here. So this is the last one we're doing today. Cell structure, cell diversity, diffusion, osmosis. But then everybody, look at this. The week of May the 10th. We're going to do those 5 big chapters, okay? And do you know, that's Sunday the 10th, 11th, 12th, 13th, 14th. And then we're doing another biology blitz on another 40% of your paper on the question 8, 9, and 10, the 3 experiment questions. And the Wednesday the 27th and Thursday the 28th, I'll be doing Unit 1 again. So that's another big one. Now, on top of that, everybody, I am doing May the 10th in Cork in person. All right, so I'm going to be doing that in Cork in May the 10th. And I'm going to be doing in the Talbot Hotel, I think the 25th and the 26th. Even though I'm doing the 25th and 26th those evenings, I'm also going to be doing 2 in-person ones those days as well. This is all going to be, you're going to see all this over the next week or so. And also the week after this, see this week here, the 24th to the 28th. The week actually before that, so the week of the 18th, basically the week between those 2 biology blitz. See, between those May the 10th and May the 24th, in between those 2 biology blitz, I'm doing another week of 5 classes, 5 days in a row. So look, you'll see it all. I'll show it you all the time, but we've so much to do. I'm also going to be doing the Monday night before your leaving cert, for anybody who's got questions, and I'm also doing that Saturday morning, the weekend before your biology exam. So there's lots to look forward to. Anyway, let's get going today, everybody. As I said, the notes are on the news feed. The notes are on the admin live chat as well. But what we're going to do today, as I told you, is we're going to do cell diversity first, then we're going to do cell structure, and then we're going to do diffusion osmosis. These are short chapters, so we're going to get through these a little bit quicker. Okay, and as I said, that should be then 30 to 40% of your leaving cert, maybe a little bit more. And don't forget, before I leave this document, question 8 has to be on those 8 experiments. Question 9 has to be on those 7 experiments. Question 10 has to be on those 7 experiments. So, and you have to do two of those, and that's 7.5 and 7.5, 15% of your paper. Let's get going today. So, if we open up, you know what I'm going to do first? I'm going to go with cell diversity. I'll tell you the reason why. Cell diversity hasn't been asked properly by itself since 2019. Now, it came up a couple of years ago as a tiny part of a long question, like 6 marks. But it hasn't come up as a short question in a long time. And as you can see here, look here, 2010, 2012, 2016, 2019. So it hasn't come up as a as a question by itself since 2019. So hopefully 2026, it'll be maybe 20 marks. You never know. So this is a really short one. It's not going to take very long. Let's go, but we don't want to take it for advantage. We got to know it. So let's get going here. Sean Mack, what am I covering in Cork? I'm going to be doing genetic crosses and DNA and RNA in Cork again. Because I think it's a good one to do in person. But anyway, you'll see all that in the platform coming up and advertised now over the next few days. So you'll see all that. So look, I think I sat down between this biology blitz I'm doing this week and what I'm doing then for the month of May. I think we'll pretty much do the majority of the course again. All right. So because we're doing sometimes a class most days or sometimes 2 classes in a day, and you can pick and choose which ones you want to come along to. So let's have a look at this. What is meant by a tissue? It's a group of similar cells that carry out the same function. Now that's a simple definition, but you got to know it. And definitions make up 15, maybe even more, 20% of your leaving cert, either directly or indirectly. All right? Because sometimes if you know a definition, it helps you answer a form of question. So if you then look at this here, okay, are you uploading these videos on feed? Oh, no, all of these videos are on my, I better show you this here. Look, before I go on any further, in case I forget. So look, the videos that we've done this week, all right? This is obviously for the members here. So the videos we do this week, you'll see here. So see where it says master classes. All right, go across the whole way. These are all the master classes I've been doing every Sunday night. But keep going across and you'll see then, there's enzymes I've done. There's respiration. There's cell division. There's photosynthesis. So there's the 4 classes we've done this week. Okay, so, and today will go up there as well. So it's just go under master classes and you'll see it there. Okay, brilliant. Thanks for asking that. All right, now, that's what a tissue is. Now, you need to know 2 plant tissues and 2 animal tissues for this chapter. Now, you're going to learn 4 types of plant tissues when you study the plant chapters later on in the course. You'll study many types of animal tissue, but in this chapter, they can only ask you to go for name 2. Okay? So, if you look at this here, you will see, all right? We study all 4 of these when we study plants. We did this actually plants about 3 weeks ago. We did all this. But in this chapter, I'm going to pick these 2. I'm going to go for dermal tissue and I'm going to go for vascular tissue. They're the 2 that we're going to name. All right. Now, what you need to know for those two. Well, let's go over the next page, or the bottom of that page. Dermal tissue, you see it there. This is a root, okay? So this is a root of a plant. And I'm sure you recognize this from the plant chapter. So I'm not asking you to worry about drawing that diagram today, because we studied that diagram in the plant structure chapter, and we've already done that a few weeks ago. But the reason why I have it here is because I want to show you that dermal tissue is always found on the outside of a leaf, a stem, and a root. It's always the outside tissue. Now, what's its function? It protects the plant. Now, you then need to be able to give an adaptation. How is the dermal tissue adapted to protect the plant? It has thicker cell walls. In that, the cells in that tissue have thicker cell walls to help with protection. Okay, so that's the first one. Remember, we have to name two. So then we're going to go for vascular tissue. Now, as you know, I'm sure you know this already if you've studied plants, there's two types of vascular tissue. You can choose whichever. So let's go for xylem. Xylem transports water and minerals, okay, through a plant. We know this because we studied this in the plant structure chapter 3 weeks ago or 4 weeks ago, whatever it was now. And we know the xylem and the phloem are found in vascular bundles. We're not going into that kind of detail today. We're just asked to name two plant tissues here and their functions. So we said xylem allows for transport water and minerals. It also provides strength because it contains lignin. So we're just going to note here, how is xylem adapted? Xylem is narrow, so water can pass through up against gravity more easily. Xylem has pits. These are little openings that are found in the xylem wall, so they can go from one xylem cell to another. And xylem has lignin, which gives it strength, so the xylem tissue doesn't collapse inwards, so water can move up. As I said, we study all of those when we study plants. For this chapter, know the name of the tissue, know its function, and know one adaptation for this chapter when it's asked. Okay, so there are your two tissues. We're going to go for dermal tissue, and we're going to go for xylem. Now, you could have went for the other ones as well, the meristematic tissue, ground tissue, or you could look at the other type of vascular tissue, ground phloem. But I'm going to go with those two because that's all they can ask for in this chapter. So I'm going for dermal tissue, and I'm going for the vascular tissue called xylem. Okay, so before we look at the next one, let's look at some exam questions they've asked in this. So if we go and look at this here, so look at the bottom of page 42 from your question bank. When this chapter comes up, it's always the same questions that are asked. Okay, so if you look at this here, the definition of tissue, it's a group of similar cells that carry out the same function. We saw that, right? Now what they can ask you is, name two types of animal tissue. Sorry, I'm going to go back to animal. We'll do that one later. We're going to go question 3. Name two types of plant tissue and state their function. Well, xylem or dermal tissue protects the plant, and xylem transports water and minerals through the plant. So that's what you need to know for this chapter. If this chapter's asked, that's what they'll ask about the plants. Now, they'll also ask, how are both plant tissues adapted to carry out their function? So how is xylem? Xylem has cell walls that are thickened. So thickened cell walls to help with protection. That's dermal tissue. Xylem, on the other hand, xylem is narrow. And there you go. That's how, and that helps the water move up through the xylem because it's narrow and it's moving up against gravity. Or it has pits. So they're the ones you have to go for. That's, if you know that about, name your two types of plant tissue, give the functions of both plant tissues, and state how they're adapted, then you know what you need to know for this chapter. Now, you need to know a lot more about those plant tissues for other chapters, but in this chapter, that's what they'll ask, okay? Now, if we then go back here, we're then going to look at remember we said we have to know two plant tissue, but we also need to know two animal tissues. So see here, we need to know two animal tissues. So I'm going to go for these two. So I'm going to go for muscular and nervous tissue. They're the two I'm going to go for. Now, you might have other ones. You might have studied other ones for this chapter. But for this chapter, they're the two I always study because we study them in great detail in the nervous system chapter or in the skeleton chapter, okay, where we study muscles and the nervous tissue. But let's go for the nervous tissue. You can see I've given another one there as well, but you only need to look 2, so I'm going to go for 2. So I'm going to go for nervous tissue. Now, what's the function of nervous tissue? Well, nervous tissue, and this chapter, the nervous system, as one of the chapters I'm going to be doing in my next biology blitz, but nervous tissue is composed of nerve cells called neurons. That's what nervous tissue is. But what's the function? It carries electrical impulses to and from the brain. So that's what nervous tissue does. Now, how is nervous tissue adapted? Now, we draw motor neurons, sensory neurons, interneurons. We actually draw this diagram in great detail in the nervous system chapter. I'm not asking you to draw it or label it for this chapter. I have it here for a reason, because look, I'm going to go for red here. So imagine now the electrical impulse traveling through the neuron. See it in red there? See it red? So the electrical impulse is traveling through the neuron. How is that neuron adapted for the transmission of that electrical impulse? The myelin sheath. The myelin sheath insulates the movement of the impulse. So that means then the impulse can travel at a faster speed. Okay? So that's how your nervous tissue is adapted. The myelin sheath insulates the electrical impulse. So that's that one, okay? Now, what about another one? You need to know two animal tissues. So the other one we're going to go for is muscle tissue. Now, what's the function of muscle tissue? Now, muscle tissue allows for movement by contracting and relaxing. Okay, so it allows for movement. Now, how is muscle tissue adapted for that? Well, muscle tissue has large numbers of mitochondria to provide the necessary energy. Now, I like this one. I think that one's an easy one to remember compared to the nervous tissue one, although you still need to know it for the nervous system chapter. But I think muscle tissue allows for movement. All right, how is it adapted for that though? It has large numbers of mitochondria to provide the necessary energy, because muscle tissues require a lot more energy than a lot of other types of tissues in your body, so therefore it has a large number of mitochondria. And an example, of course, is your skeletal muscle, your bicep and tricep. Let's have a look what they can ask on that then. So this is what they can ask on this. Look here. So we'll go back here. So if you look at question 2 on the bottom page 42. So here it is. Name two types of animal tissue. So muscular tissue and nervous tissue, and state their function. Muscle tissue allows for movement. Nervous tissue carries electrical impulses to and from the brain and spinal cord. So that's the functions. Now, and this has been asked in 2019, for example. 2019, this is what they asked. Okay, the last time this chapter was asked properly as a chapter by itself. Now, if you then look at part 2, state how each of these tissues is adapted to carry out the function. Well, how is muscle tissue? Muscle tissue has a large number of mitochondria to provide the necessary energy. Okay? And nervous tissue, we said the myelin sheath insulates the electrical impulse. So that myelin, I'll go back here and show you that again. So the myelin sheath insulates the electrical impulse. Okay, you see it right there. Now, both of them in 2019, you had to be able to name two animal tissues, and you need to be able to give the functions, the adaptations. So sometimes they do ask for the two, and that's why you need to know for the two. And that's all they can ask for in this chapter so far. Two plant tissues, two animal tissues, state the function, state their adaptations, and there you go. There is other adaptations as well, of course, but they're the ones I go for. Now, let's go to the bottom page 89. The second, now that's half the chapter done. So now we're looking at the second half and say this is a very short chapter. Okay, so what we need to know then is we need to know what the word tissue culturing. Tissue culturing means what? It's growing cells in a sterile nutrient medium outside an organism. So let me give you an example. We can take skin cells from a person's skin. We can grow those cells in a sterile nutrient medium. So that means we can grow those cells in an environment free of microorganisms, and we can provide those cells with nutrients, and those cells will then grow outside the organism. So let's say someone suffers from a skin burn, maybe from a fire or from whatever, right? And that person needs a skin graft. Well, generally what they do is they take skin from another part of their body, let's say their leg, and they place that skin then on the, let's say if it's on their face or their arm where the skin was damaged by the burn. Now, what they can do now though is this. They can now grow skin cells in a sterile nutrient medium outside the organism, so they can take skin cells from that person's body, healthy skin cells. They can grow them in a lab in a sterile nutrient environment, okay? And those skin cells will then divide by mitosis and form new skin cells. Now, you have to give the environment, the right environment for those skin cells to do that. but we can grow skin cells, and then therefore we can use the skin cells that we grew in the lab to use as the skin graft. Okay, so if you then look at this here, so what do we mean by the word sterile? See the word sterile there. Sterile means free of microorganisms. You need to know that. And the word, you'll see the other word here, sterile nutrient medium. So the nutrient, see here, the nutrient is the food that we're going to provide the cells with. So that's what we mean by that. A lot of people learn that definition don't grasp what it means. You're going to grow cells, maybe skin cells, maybe plant cells, in a sterile nutrient medium. So an environment that's sterile, which means it's free of microorganisms, so the skin cells can grow without worrying about other microorganism growth, and we provide them with food, and they'll grow outside the organism. All right, skin cells would normally die if you didn't give them the right environment. But if you can control the environment, then they'll grow outside that organism. So the word in vitro, we often refer to in vitro like IVF, outside. So when you're growing cells outside the organism, we say it's in vitro. Now, if we then go down a little bit further, it's not just skin cells from animals, from humans we can do this. We can grow plants like this as well. So what you basically do is we take cells, not a seed, we take actual plant cells. You give these cells an environment, a growth medium, so that's nutrients, okay? A growth medium is the nutrients. So we provide these plant cells that we've taken from a plant, and you provide them with a growth medium, nutrients, and they will divide, they will multiply. And what you will have then is, you'll have then lots and lots of small little plants growing that are all identical to each other. Why are they all identical? Because all of these new plants grew from cells, not seeds, actual cells that came from one parent plant. Okay? So let's have a look at this. If we want to grow skin cells to make a new piece of skin, or if we want to plant cells that divide and multiply into new plants, okay, what do we need to do? Go to the next page. We must provide them with certain factors. So if we want skin cells to grow outside our body in a lab environment, if we want plant cells to grow outside that plant, take plant cells from that plant and grow those plant cells in an environment like a lab environment, how are they going to grow? Well, you first of all have to give them the right nutrients. You also might have to make sure you provide them with a suitable temperature, a suitable pH. You also need to provide them with oxygen. All right? You also need to make sure the environment is sterile, free of microorganisms, because we're trying to grow skin cells or plant cells. Okay? Remember, for those that are not usually on the platform, these notes are on the live chat, but they're also on the news feed. So don't forget that. Now, if you then look at this here, why do we want to make sure it's sterile? We want to make sure we don't have any contamination, because if bacteria grow in that environment that we're trying to grow the skin cells, then them bacteria might kill the cells that we're trying to grow. Okay? So really important that. Now, let me just go and show you a couple of exam questions they can ask on that. This is a short chapter. There's not much left to it. So if we look at this here, look at this here. Question 5 on page 43 on your question bank. Okay, so if you look at this here, you will see here, define the term tissue culture. Tissue culture is the growth of cells in a sterile nutrient medium outside the organism. So that's the definition, right? It says, name 3 factors. Now, in your Leaving Cert, I've only ever seen 2 being asked, but I said 3 just in case. Name 3 factors that are needed for a successful tissue culture. You must provide the cells with correct nutrients. You must provide them with a suitable temperature or a suitable pH. You must provide them with oxygen supply. Highlight 2 or 3 of those so they're easy to remember. Okay? Brilliant. Let's now go back and look at the last thing, one of the last things here on tissue culturing. We need to know where we can use tissue culturing, the application of tissue culturing. We need to be able to give a couple examples. So I'm going to go for these 2. Skin grafts and micropropagation, because we need to study micropropagation in a chapter called vegetative propagation in the one of the last plant chapters, which I'll be doing next Thursday, by the way. So let's have a look here. Micropropagation and skin grafts, or you could say cancer research, but I'm going to go for these 2 in a little bit more detail. So micropropagation, what is that? It's where we're growing large number of plants from cells. So look what we're doing. You're just like back here, see back here, you're taking cells from the plant. You grow these cells in the environment that will allow them correct nutrients, suitable temperature, suitable pH. And these plant cells or animal cells, could be skin cells, whatever it is. Well, in this case, it's micropropagation, so it's plant cells. These cells will grow. They'll divide by mitosis and they'll form a callus, a clump of cells. So, here's a carrot. Imagine this carrot is the perfect carrot. It tastes good. It's got the right shape. So everything that the farmer is after to sell to the market. So what we need then is you want to produce lots more carrots like that, identical to it, genetically identical. So you take cells from that carrot. These cells, you provide them with the correct nutrients, suitable temperature. Those cells then will divide by mitosis to form a clump of cells called the callus. And that will then continue to divide by mitosis until you get a brand new plants. And those new plants are identical to the original because those cells just came from one plant. Okay? So that's what micropropagation is. It's where we grow plants. It's where we grow a number of plants from plant cells. Now, if we go here, what's the benefits of doing this? Why would you want to grow plants this way? Why would you take cells from one plant, provide them with the correct nutrients, provide them with a suitable temperature, provide them with oxygen, whatever they need? Why would you want to do this, to grow plants this way? Well, you can produce a large number of plants in a very short period of time. Okay? And they're all genetically identical, which can be positive because then they'll all have the same characteristics, the same taste maybe, the same look, whatever it might be. So that's the first one. The other application of tissue culturing is skin grafts. I think people probably prefer this one. So skin grafts, it's why would you want to grow skin cells outside an organism, all right, in a sterile nutrient medium? Because people might need skin grafts to replace damaged skin from maybe a bad burn or whatever it might be. But rather than taking that skin from somewhere else in their body, they can now grow new skin cells that will not be rejected because these skin cells that grew in the lab, these cells came from that person's body. You took some skin cells from that person or some cells from that person and then you grow them in the lab. So your body recognizes these cells as your own. All right. And it also means you don't have to remove skin from other parts of your body. Okay. Brilliant. So let's have a look at this then. Applications of tissue culturing. So look here, question 7a. Look at question 7a on your question bank. Page 43. Give 2 applications of tissue culturing: skin grafts, micropropagation. Okay? Now, if you then look at this here, state a benefit of each of these applications. What's the benefit of growing plants like this? You can produce a large number of plants in a short period of time because you're growing these plants just from cells that came from one plant. Also, the benefits of skin grafts means the person's body will not reject that skin, even though that skin was not taken from that person's body, all right, from another part. The skin cells that grew in the lab, your body recognizes them as your own because the original cells that piece of skin grew from came from that individual body. So, look at this here. Name the type of cell division involved in tissue culturing. It's mitosis because one cell divides into 2 identical cells. So that allows for growth to occur. Brilliant. The last little thing in this chapter, there's not much left. You need to know these 2 or 3 definitions and a couple examples. So let's have a look at this. So you see, so Sophie, you said, what do you mean by medium? A growth medium is the food, the nutrients. Okay, we mentioned that. Look here. So the growth medium, you'll see it there. There's the growth medium there. So the growth medium, where are we there? Back, I've gone too far now this time. So a growth medium is, you'll see there, let me get my pen here. Here we go. So a nutrient medium. So that means you're providing the food for the cells to grow. Now, if you look at this, everybody, look at this. What do we mean by the word organ? An organ is a number of tissues working together to carry out one or more functions. Now, don't get these definitions mixed up. So the first definition was a tissue. A tissue is a group of cells carrying out the same function. A group of similar cells that carry out the same function. But an organ is this. An organ is a number of tissues working together to carry out one or more functions. One or more functions. So if you look at this here, a lot of people don't realize that plants have organs. So the root is an organ. Now, if the root is an organ, if the root is a plant organ, that means, let me just show you this here. If the root is a plant organ, that means the root is made up of a number of tissues. Yes, it is. The root is made up of dermal tissue, vascular tissue, and ground tissue. I showed you this. We studied this in the plant chapter in more detail, but I mentioned it briefly. Look, there's the root, and see all the tissues that make up that organ of the root. So that's why the root is an organ. If you look at then the second plant organ, the leaf is an organ because it's also made up of a number of tissues that carry out one or more functions. So the leaves are made up of dermal tissue, vascular tissue, and ground tissue. They all carry out different functions. So that means the leaf is an organ. Okay, what about an animal? Give me examples of animal organs. Well, let's go for the heart. The heart is an organ. So hold on now, everybody. If the heart is an organ, it means it's made up of a number of tissues that carry out one or more functions. Look, so the heart is made up of nervous tissue, because that tissue sends out electrical impulses, causing the muscle of the heart to contract. The heart is made up of cardiac muscle, and the heart is made up of connective tissue. So that means the heart is an organ. A lot of people just learn that definition, but then they can't apply it to what we're saying. If the heart is an organ, that means it's made up of a number of tissues working together to carry out one or more functions. And you see there, the heart is made up of nervous tissue, connective tissue, and cardiac muscle tissue. Okay, that's what we need to know about that. What about the last thing here? You need to know what an organ system is. So what's an organ system? I know that's another definition. So what's a system? It's where you got two or more organs working together to carry out one or more functions. Let's have a look at some of our systems that we study on the course. The digestive system. Now, if the digestive system is a system, it's made up of two or more organs. Well, let's just prove it. The digestive system is made up of the mouth, which is an organ, the esophagus, the stomach, the intestines, the large intestine, the small intestine. So that means that's two or more organs. So you see there, two or more organs working together to carry out one or more functions. So 1, 2, 3, 4, 5. And more of them as well. But there's 5 organs that make up your digestive system. And all of those organs carry out sometimes similar, but sometimes different functions. Okay? Perfect. And look at another system. Another system is the respiratory system. If it's a system, that must mean it's made up of 2 or more organs that work together to carry out one or more functions. So the respiratory system, that's your breathing system. It's made up of your mouth, your nose, your trachea, your alveoli, your lungs. These are all organs that make up that system. So there you go. So I want us to go back and highlight or to look at 2 or 3 more questions on this. And then if you know these questions, then you can say you know this chapter. And this chapter has not come up by itself since 2019. So if you look at this here then, so look, define the following terms. An organ is a number of tissues working together to carry out one or more functions. What's a system? 2 or more organs working together to carry out one or more functions. Now, finally, look at this here. Name 2 tissues in an organ of the heart. Well, the heart is an organ. So what 2 tissues? The nervous tissue, muscular tissue, connective tissue. Now, I've said 3, but they only asked for 2. So you only give 2. I'm giving 3 because we're revising today. Name 2 tissues that are present in a plant organ of the leaf. Well, the leaf is an organ. It's made up of dermal tissue, vascular tissue such as xylem or phloem, and ground tissue. And finally, name 1 example of an organ system in humans. Well, the digestive system. Name 2 organs that feature in it. Well, the mouth, the esophagus, but there's many others. So you got to be able to not just learn the definition, but be able to understand what the definition means in relation to what them questions there. So, Sophie, you said, do you think this could come up as a short question? Sophie, this has not come up since 2019 as a question by itself. Now, it appeared a couple of years ago as part of a long question where it was like 6 marks or maybe 9. But it hasn't come up by itself since 2019 as a short question. So look, there is a chance. That's 7 years ago now. It was 2019, 2012, 2010. Those are the 3 years it was asked as a short question. And in 2016, it made up half a long question. 30 marks, 7.5% of your paper. Okay, that's that one, everybody. We're going to now go and do another short chapter called cell structure. Remember, this is the plan. So look. So we've done, let me show you here now. Here we go. So look, we've done, here we go. So we've done now cell diversity. In the next half hour, and then we'll have a 5-minute break. In the next half hour, we'll do cell structure. Okay, and then in the second half, we'll then obviously do osmosis diffusion. But if you look at cell structure, everybody, I just want you to note here, the cell structure chapter has two experiments. So there's 2 experiments in the next chapter we're doing, and those 2 experiments could come up as question 8. So let's go and have a look at this chapter. Now, if you look at this chapter, we're going to go, and we'll go back here now. So we're going back to page 72. Now this chapter came up last year as a short question. Okay? Then it didn't come up for 3 years, for 7.5%. Alright? But then sometimes it comes up, sometimes you see here 1, 2, 3, 4, 5 years in a row. So it's hit and miss, this one. But my point is, this is the point I'm trying to make here. You need to know all 7 chapters if you want to guarantee that you're going to get that 30 to 40% of your paper. Okay? So we don't want to leave any of those chapters out. So let's now go and look at cell structure. Now, if we look at cell structure, the first thing I want to show you is this. Let me go back and show you this. If you look at the 2 experiments, the first experiment is to be familiar with and to use a microscope. Most people don't even realize that that's an experiment. Everybody knows to prepare a plant cell and an animal cell is an experiment. But as an experiment goes, you need to be able to label the microscope, give the functions of the microscope, and describe how to use the microscope, because that's counted as an experiment. Okay? So let's go and have a look at that. If we go there, you will see on page 73. So on page 73, we are first of all going to look through the microscope. So the microscope hasn't been asked in a big way in a while. So we want to make sure we know this, and we're going to tie in then the experiments with that. Now, the first thing I want to note there is this. The eyepiece, what you see here now, and there's a few little things on this that can be tricky. So I'm going to show you the eyepiece and the objective lens. They are both lenses. So I need you to note what both do. You'll see this on the next page, page 74. So the eyepiece magnifies the image. The objective lens magnifies the image. They both magnify the image. Okay, now that leads me then, go back up to the top, page 74. So the eyepiece, you see here, the eyepiece has a magnification, let's say, of 10. It magnifies the slide 10 times. The objective lens magnifies it 40 times. So how do I get total magnification? What do I do to the 2 numbers, everybody? So what do I do to the 2 numbers? I'll give everybody a second there. So what do I do to those 2 numbers? If I want to get total magnification, what do I do to the 2 numbers? Okay, brilliant. So what I do to the 2 numbers is, of course, I multiply them. So I want you to note there, how do you calculate the total magnification? Now, but here's the thing. They asked it like this. Was it 2016, I think it was, or was it 2018? They asked this. How do you calculate total magnification? And a lot of people say, oh, you multiply the 2 numbers. But they couldn't actually put it into writing, though. So, yeah, you multiply the 2 numbers. But this year, they asked you to put it into writing, though. So how do you calculate total magnification? You multiply the magnification power of the eyepiece by the magnification power of the objective lens. So that's how you put it into writing. And that's what they wanted that year. You multiply the magnification power of the eyepiece and you multiply the magnification power of the objective lens. So in this case, 10 by 50, 500. Okay? Or 10 by 40, 400. So that's what you need to know about the eyepiece and the objective lens. Okay. If we then look at the next part, how do we then move the objective lens? Sometimes you might have this objective lens, and then you might have this, you want to move to the other objective lens. So how do you switch from objective lens to objective lens? You use the nose piece. So let's just note there, the nose piece allows you to move the objective lenses into place. Excellent. Now, what then? The stage and the clip. Don't take them for granted. I know they're pretty obvious, but the stage holds the slide, but the clip holds the slide in place. Okay, brilliant. There's not much left here. What next? We're going to go then for the coarse of focus and the fine focus. Now, while I give the function of each of those, which one should you generally use first? The coarse focus or the fine focus? Let's look at the function while you might tell me which one you use first. The coarse adjustment is for rough focusing. All right, so it's for rough focusing. So it allows you to detect what you want to see. Let's say a plant cell or whatever it may be. But then you want to maybe magnify it and you want to make it more precise. Then you use for the fine focus. The fine focus is for more precise focusing. Okay, excellent. Now, there's only 2 more parts left. And the diaphragm is a really important one. So the diaphragm, look, you see where the diaphragm is? The diaphragm is this part here. Okay, now what does the diaphragm do? It controls the amount of light entering the slide. It controls the amount of light entering the slide. So you don't want, imagine there's the slide there, okay? You don't want too much light passing up through or too little light, because then it's not going to make the slide and what's on the slide visible. So you want the right amount of light. Okay, now I've never seen this one being asked, the condenser. The condenser is just above the diaphragm. Okay, generally when they're pointing underneath there, it's usually the diaphragm. But if it clearly points to the condenser, then what does the condenser do? It focuses light onto the objects. So the diaphragm controls the amount of light. So let me just, here we go here. So the diaphragm, here we go. So the diaphragm controls the amount of light reaching the slide, but the condenser focuses it. So it's nicely focused onto the right part of the slide. Now, what do we know so far? We need to know how to label the microscope. Remember, this is one of the experiments. It could come up as an experiment, right? You need to know, there's rumors out there that, and sometimes I think this as well, you should know all the experiments, but I think there's a slight chance that one of these cell structure experiments could be asked this year. So know how to label it, know the functions of the different parts of the microscope, but then know how to use it. And how do you describe how you use it? You'll see it right here. Bottom page 74. Now this is very poorly answered in your Leaving Cert. Very poorly. And it's been asked about, I think, 4 times. 4 times, because there's the plant cell experiment, there's the animal cell experiment, but then there's another experiment using a microscope called the transverse section of a diacot stem, where you cut a thin section of stem and you place it onto the slide, and onto the stage of a microscope. So this could be asked for the 3 experiments. Describe how you use the microscope. Now, you place the slide on the stage and you clip it into place. That's probably not going to get you marks. It just depends. So I would always write it in, no. Switch on the light source. Probably not going to get your marks, but you just never know. Write it in. But this is where you normally get your marks. You use the coarse focus with the low power objective lens. So look here, you use the coarse focus. So use the coarse focus there, see that one there? With the low power objective lens. And then what you do, I'll go to yellow, you then switch to the fine focus and you switch to the higher power objective lens, whichever one they might be. So I want you to note there, so you use the coarse focus with the lower power objective lens. And then when you see what you want to see, but you want to magnify it even more, you then switch to the higher power objective lens. So you move the higher power objective lens into place. So you move the higher power objective lens into place. And then you use the fine focus. You then use the fine focus. And that's how you use a microscope. Okay? So I'm going to ask you to do a couple of questions on this. So I want you to turn to your question bank, and I want you to turn to question, you see it here, page 36, question 1. Now, I'm not going to go through all these, but you need to know the functions of all the parts. So I'll just pick the objective lens and the eyepiece are both lenses. They both magnify the image. The diaphragm controls the amount entering the slide. I won't go through the rest because I think they're pretty straightforward. You need to be able to label the microscope. So I'm not going to go through that again. It's pretty straightforward. Okay? What then? You then put a star. This is a really important one. You need to be able to describe the steps involved when using a microscope to view an object on the slide. So how did you use the microscope? You placed the slide onto the stage and you clipped it into place and you switched on the light source. As I said, that's probably not going to get marks, but it could. Marking schemes do surprise you from year to year. No harm to throw it in. But this is the key point. You use the coarse focus knob with the lower power objective lens, and then you switch to the higher power objective lens and use the fine focus then for more precise focusing. So that's really important. That does get asked a few times. Now look at question 4. When using a microscope, describe how you calculated the magnifying power of the microscope. I think that was 2016. Or was it 20? It was around then. So how do you calculate the magnifying power? You multiply the power of the eyepiece by the power of the objective lens. That's what you do. When you see it in writing, then it's pretty obvious. But a lot of people say multiply, but they don't know what they're multiplying. You're multiplying the power of the objective and the eyepiece lens. Now look at another one. All right. How do you alter the amount of light shining onto the specimen, onto the slide? You use the diaphragm. So the diaphragm is the answer there. Now this was another question, question 5. Describe the steps taken to change the magnification. This was asked, I think, in 2022. Describe the steps taken to change the magnification from low power to high power. Okay, so how do we go from low magnification to high magnification? Place the high power objective lens into place and then use the fine focus knob to focus it correctly. So you're going from a lower power magnification to a higher power magnification. So you place the higher power objective lens into place and you use the fine focus knob to focus it correctly. Perfect. Now, what we're going to just note there's a couple other things they can ask you about the microscope. Okay? And I want to make sure you get all of these little questions. So this is what they can also ask you about the microscope. They can ask you to calculate the actual size of an object. Okay, so this has been asked twice in 2016 and 2022. Okay, so if you're asked, now you might use the triangle if you prefer, or you might not. So look, that's what these letters represent. So let's go for A. Okay, if you want to calculate the actual size of the object, let's say you want to calculate the actual size of the cell that you're viewing onto the microscope, you get the image size and you divide it by the magnification. Now this one has been asked twice in your Leaving Cert. This one's come up twice. Okay, so please highlight that one. The actual size of the object, how do you find it? You find the image size and then you divide by the magnification. Because the image size is when it was magnified. So when you magnify it, let's say 300 times, that's its image. But then when you want to find its actual size, then you want to shrink it back down to what it was originally. Okay, before you magnified it. Let's put one of the two of these into action then. I want you to look at this one here. In a microscope, the objective lens, and as I said, 2016 and 2022. Or was it 2014? It was around then, okay? This is when this type of question was asked. If the microscope with an objective lens has a magnification of 30 and the eyepiece has a magnification of 10 and it created an image that had a diameter of a plant cell 0.9 millimeters. What's its actual size? So let me go show you this here. So what was its actual size? Now, if you look at this here, how do we calculate the actual size? The actual size, hold on, I get my pen here. The actual size is the size of the image. So the size of the image, look, the image was 0.9. So the size of the image was 0.9. Divided by the magnification. You divide it by the magnification. Now, what was the magnification? It was magnification 30 and magnification 10. So how do I get total magnification? 10 multiplied by 30, which is 300. So what I use that number now for? I say the image, remember we said, let me just go back up here. Look here. You divide the image by the magnification. So image size divided by magnification. So the image was 0.9 and the magnification was 300. So 0.9 divided by 300, you get 0.003. Okay? And that's the answer. And that's come up twice in your Leaving Cert. Not that exact numbers, but that type of question. I'll do another one. Okay? So if you look at this one here. So, no, actually, I'll show you the one. I'll show you the one on the next page here. Actually, on the question bank. Let's do the one on the question bank. All right. Let's have a look at this one. So the one on the question bank is question 9 here. Look at question 9. So question 9 states, well, that's the one we just done. So actually we'll go to question 10. So that question 9 is the same as the one in the notes. We'll look at question 10. Question 10 says, an eyepiece has a magnification of 15 and an objective lens has a magnification of 50. So before we go any further, total magnification, you multiply the two and it's 750. Okay? It's 750. Okay? Now, what then? If the image is 2 millimeters, what's the actual diameter? So do you remember what we said? The actual size is the image divided by magnification. The image, they tell you in the question, is 2 millimeters. So 2 divided by magnification, it was magnified 750 times. So if you divide 2 divided by 750, you then find its actual size before you magnified it. And that's 0.0026. That one came up 12 years ago. That's the exact one that came up 12 years ago. Or something very similar. Maybe the numbers I might have changed slightly. But it's the same type of question. Okay, they're easy if you've done them before. But if you've not done them before, they're difficult. So all you're going to do, one more time, if you want to find the actual size of the cell that you're viewing, what's its size in real life? The image will be given to you in the question, and the magnification, you might have to multiply the two numbers of the two objective lenses, but all you have to do then to those numbers is the image divided by the magnification. The image divided by the magnification, and that's how you find the actual size. So please, if this chapter does come up and it comes up as, and the microscope comes up as one of the experiment questions, there's a decent chance that one of those calculations will be asked. Because in 2014, or was it 2016, whichever year, and in 2022, the microscope came up, and in both times, all right, and the plant cell, animal cell came up, and in both times, that calculation was asked. Okay? Now, let's move on. All right? Let's move on. So what we're going to do now, everybody, is we're finished the microscope. But we're now going to look at the plant cell, animal cell experiment. So if we now jump to the end of the chapter for a moment, let's go to question page 83. So let me just show you what we're doing. So this is what we're doing. So let me just show you here. Let me go 1 wee second here now. So if you look at this here, there you go. So if you look at this here, let me just rub that out. So we've just done that experiment to be familiar and to use a light microscope. That's what we've just done. Okay, and now we're doing this experiment to prepare a plant cell and to prepare an animal cell using a light microscope. So that's the other experiment in this chapter. All right, and a lot of people don't even, this is the crazy thing, a lot of people don't even, I'm sure some people here, until they saw this week, didn't know that question 8 was on those 8 experiments. And if you don't know that, then you're at a disadvantage, because you studied those 8 experiments, you got 7.5% of your paper. So that's why it's so important that you know which experiments are grouped with which question. Now, let's look at the plant cell. As I said, this is the experiment. Now, this does come up. All right, plant cell, animal cell. Let's have a look at the plant cell. How do we get the plant cells onto the slide? Using forceps, you pull a thin, transparent inner leaf of an onion. So we're going to go for an onion here, okay? Now, what do we do then? You then place this small strip of onion onto the slide. What do we do then? You add iodine. How? Using a dropper. Why do we use iodine? Because it stains the cell and the nucleus an orange color. Now the nucleus becomes a darker orange brown, but it stains the entire cell. The nucleus just stains that a little bit darker. What do you do then? You add a few drops of water. Why do you add the water? It prevents the cell from drying out. It prevents the cell from drying out. That's why you add water. Okay? Now, what do you do then? You add a cover slip at an angle using forceps. Now, this was asked, once this was asked and it was worth 6 marks. How did you add a cover slip? And if you said at an angle, you only got 3 marks out of 6. At an angle, using forceps, or using a pencil, or using a needle, mounted needle. Now, can anybody tell me, why do we add a cover slip? Can anybody tell me that? Why do we add a cover slip? So, Freya, Freya just asked, do I think this experiment could come up? The microscope and the plant cell, animal cell, it hasn't been asked now, I think, since 2022 properly. So look, there is a chance, but look, the ecology experiments came up last year in question 8. So the DNA came up the year before. So look, there's a chance, but you're going to know all 8 experiments anyway. So why do we add a cover slip? Now, why do we add a cover slip? So what are we saying there? So brilliant, because I thought I was going to try to catch us out there. Because why do we add a cover slip? I thought some people were going to say to prevent air bubbles. No, we add a cover slip to protect the lens. Because if the stage goes up, oh, someone said it there, to prevent air bubbles. Okay? No, you add a cover slip to protect the lens of the microscope. Because if you bring the stage up too high, then the iodine on the slide will then hit off the objective lens. So you don't want, that's why, look at this here. You don't want, if you bring the stage up too high, then it's going to hit off your objective lens. So that's why you add a cover slip. It protects the lens. But the other reason why we add a cover slip is to prevent the cell from drying out. Now, however, let me ask it a different way. Why do we add a cover slip at an angle? That prevents air bubbles. So be very careful how that question is asked. Let me go through the 3 questions. They'll ask you about the cover slip, if they do ask you. So they say, how did you add a cover slip? At an angle, using forceps. Why did you add a cover slip? To protect the lens or to prevent the cell from drying out. Why did you add the cover slip at an angle? To prevent air bubbles. So there's the 3 questions. So be careful how they ask that. Okay? Brilliant. Yeah, you can use, it doesn't have to be forceps there, Roisin. It could be a mounted needle, it could be a pencil. There's many options. Okay, now, if they ask you to draw the plant cell, you draw the basic structure of the plant cell because you're only using a light microscope. You're not going to see the finer details like the ribosomes and so on. So you just draw that simple plant cell. Okay, let's look at the other one, of the part 2 of this. To prepare an animal cell. Okay, so you rinse your mouth. You scrape the inside of your mouth with a cotton bud. Really important that. You spread those cells then, those cheek cells, onto the slide. What do you do then? You add methylene blue. It was iodine for the plant cells. It's methylene blue for the animal cell, the cheek cell. You need that. People don't know that name, and because it's nowhere else on the course. It's just on this one part. Methylene blue. Why do you add it? Same reason why you add iodine. It stains the cell and it stains the nucleus. Now the nucleus becomes a darker blue even. Why did we add a few drops of water? To prevent the cells from drying out. Again, how did you add a cover slip? At an angle using forceps. Why did you add the cover slip at an angle? To eliminate air bubbles. Why do we even use a cover slip? To protect the lens. And again, you draw your simple diagram. If they ask you to draw the diagram, you don't draw the ultrastructure. You're not drawing mitochondria. You're not drawing ribosomes. You're only using a light microscope. So you draw the cell membrane, the nucleus, and the cytoplasm. Okay? Brilliant. So, before we go and do a couple other things in this chapter, all right, I just want to show you this here. So I want to show you just a couple of questions on, here we go here now. So you will see here, all right, so we're on page 40, we'll go here, where is it there? Page 41. Let's go and look at page 41. And these questions are only simple if you know them. So let's make sure we know them. So what type, we're looking at the plant cell. So what type of cell? We used an onion cell. How did you obtain it? Using forceps, you pull the thin, transparent inner leaf of the onion. All right. Why did you use a cover slip? To prevent the cell from drying out or to protect the lens. Any one of them. How did you add the cover slip? At an angle using forceps. That was 3 and 3 one year. That was 6 marks. Why did you add it at an angle? To prevent air bubbles. Okay. So name a stain you use to view a plant cell. Iodine. What's the benefit of using that stain? It makes the cell more visible. It makes it easier to see. Okay, it stains the nucleus. It makes it easier to see. You draw your diagram of your plant cell. Okay, perfect. Let's look at the animal cell. Look at this one here. This is now based on the animal cell. What type of cell did you look at? A cheek cell. How did you obtain the cheek cell? You scrape the inside of your mouth with a cotton bud. Again, look at this here. You will see here, name the stain that you used. Methylene blue. How did you apply the stain? Using a dropper. Why do you apply the stain? To make the cell more visible or easier to view. The reason why these questions are here, because they've all come up in your leaving cert, high level papers. Okay? Which organelle is stained most strongly? It's the nucleus. Look at this question here. Alright, question 66. Describe the difference in color, in depth or depth of color, between the nucleus and the cytoplasm when you viewed the animal cell. The nucleus is a deeper blue, or if it was a plant cell, it would be a deeper orange. The cytoplasm is the lighter blue. Again, you draw your simple diagram. Perfect. Brilliant. Now, let's go and look at the rest of the chapter. And there's not much left in this chapter. There's not much. But there is a few things that are important. So let's now go back to page 78. Okay? Let's go back to page 78. And I'm going to just get a quick sip of water here. Get my glass. Okay. Now, let's go to page 78 here. So, if you ask that question, Michael, at the end, I'll answer it for you. I just want to stay focused on what we're doing here, Michael. But if you ask me that question at the end, I'll give you an answer, okay? All right, let's have a look here, everybody. The structure of a cell membrane. So now, what we're looking at here, all these organelles that we're looking at them using an electron microscope. So let me actually go back to page 76 for a second. So, if you want to see the ultrastructure of a cell, if you want to see the cell's ultrastructure, you got to use an electron microscope. You need to know that. You need to know the electron microscope allows you to view the cell in greater detail. Okay? Now, if you look at this here, this is a plant cell, not looking at it, not using a light microscope, using an electron microscope. And this here is an animal cell using an electron microscope. Now, please know how to draw those. They generally, I've never really seen them being asked to draw the ultrastructure of a plant cell or the ultrastructure of an animal cell. But this could be the first year in a high level Leaving Cert paper. You could be asked that. But what we're going to do is we're going to take each part of the cell and we're going to start off with the cell membrane. So let's look at the cell membrane. The cell membrane has a really important function. And the function of a cell membrane is, I want you to go down here, is it's selectively permeable. We're on page 78. But now let's go back up and let's look at what a cell membrane actually consists of. Now, if you're asked to draw a cell membrane, they're not asking you to draw a nucleus and a cell membrane like that. That's not what they're asking you to draw when they ask you to draw a cell membrane. What they want you to draw when they're asking you to draw a cell membrane using an electron microscope, viewing under an electron microscope, is this. Because what we're looking at here, look at this, everybody. There is the cell membrane of an animal cell. Okay? That's the cell membrane. Now, that cell membrane, you see here, we're taking one part of it and we're magnifying it. And what you see is what's under that magnification. Now, we are going to draw that now because you need to be able to draw it. You're more likely to be asked to label it, but you do need to be able to draw it. So what we're doing is we're drawing the structure of a cell membrane. So let's draw this. Okay? Now, if I ask you this, everybody, from the food chapter, I know we're not doing the food chapter today, but can you tell me, what's the role of a phospholipid in a cell? Well, you'll hopefully see then, the role of a phospholipid is it forms the cell membrane. You can see this here. So a phospholipid is a type of lipid, it's a biomolecule, and that phospholipid forms your cell membrane. Okay, that comes up, not so much in this chapter, comes up a huge amount in the food chapter. So, give a structure role of a lipid in a cell. The structure role of a lipid in a cell is phospholipids, there's your phospholipid, forms a cell membrane. Okay, and that's in your food notes. But you see it here also. So what we're looking at here is the cell membrane. Now, the cell membrane consists of proteins. It consists of a pore. Now, it also then consists of the following. It contains a phosphate head, a lipid tail. And together, we call that a phospholipid. Okay? So you need to be able to draw that. You have to be able to draw a cell membrane. Now, when you're drawing a cell membrane, you're not drawing it like that. You're drawing a cell membrane in detail. If you're using an electron microscope. And does anybody know how many phospholipid layers does a cell membrane have? Freya, I'll be doing, Freya's asking when am I going to be doing food. I've done it probably since Christmas. I've done food about 3 or 4 times. About 3 times, I'd say. I'm doing food as part of my biology blitz, the last biology blitz. The week of May the 24th. It's one of the classes I'm doing there. But Freya, but the food chapter, just let me show you, Freya. The food chapter, Freya, if you can't wait that long, don't forget the food chapter is already on the on-demand. You see there? There's all the videos on the on-demand. It's probably might even be better if you watch that instead of watching the live recording. It's up to you. You can watch either, but that's where you're going to see it. Now, let's have a look. So, well done. You got a phospholipid bilayer. So that's a phospholipid bilayer because there's two layers. Okay? So there's one layer there and there's the other layer. So a phospholipid bilayer. Okay. Now, if you then, so look everybody, name two biomolecules that help form a cell membrane. Phospholipids and when I put that in there, you see here, the other one is a protein. Sorry, it was there, but the box was made smaller, so it's hiding there. So there it is. So that's your cell membrane, everybody. That's what, if you took one of your skin cells and you viewed the skin cell and you looked at the cell membrane of that skin cell and you magnified it, that's what it looks like. Or more simplified version right there. Okay? That's what your cell membrane looks like. Now, this came up a few times. What's the advantage? So a cell membrane is selectively permeable. It controls what substances enter and leave the cell. The cell membrane also retains the cell content. Very important. Now, what's the advantage of being selectively permeable? It allows the useful substances to enter. Or you could also say it keeps the harmful substances out of the cell. Now, you were asked once in your leaving cert this question. Where is a cell membrane in a plant cell? Where is it located? It's located immediately inside the cell wall. So let's just note that there. It's located inside the cell wall of a plant cell. So that's where a cell membrane is located. Now, in an animal cell, it's the outside layer. But in a plant cell, it's located immediately inside the cell wall. Okay, so before we leave the cell membrane, let's look at the exam questions that have been asked on the cell membrane. I'm going to ask you to turn to page 38. Let's turn to page 38 here. Okay, now, we'll go to question 38, or page 38. Now, if you look at this here, you will see question 14. Now, question 14, here we go here. It says, draw a label diagram to show the ultrastructure of a cell membrane. We just drew that earlier. So, you got your phosphate head, your lipid tail, you got your protein. Okay, and that there is a phospholipid, and that there is a phospholipid bilayer. They generally don't ask you to label the bilayer, but just in case. Now, if we then go down here, this is a more recent question. It says you're looking at the ultrastructure of a cell membrane. So if you're looking at a cell membrane like that, you're using an electron microscope. Okay, so what's X? X is the phospholipid. It's the phospholipid. Y, what's Y? Y is the protein. Brilliant. There it is. Excellent. Now, look at this. What's the function of a cell membrane? It's selectively permeable. Now, remember, before I leave that, what's the structural role of a lipid in a cell? Phospholipids form cell membranes. That's the link between those two chapters. All right, so look, what's the structural role of a phospholipid in an animal cell? It forms a cell membrane. Okay, now, if you then go here, let's go back here now. We're now going to look at the next organelle. Now, the next organelle we're going to look at is the mitochondria. Now, people always forget this. Plant cells have mitochondria, or mitochondrion for one. Plant cells carry out respiration, just like animal cells. All right, people forget that. All right, so they have mitochondria. Animal cells also have mitochondria. So what do we need to know about mitochondria? We need to know they're the site of respiration. So we need to highlight that. Now, the mitochondria has a double membrane, meaning it too is selectively permeable. So the mitochondria is obviously inside our cells, but even being inside our cell, it controls what enters and leaves the mitochondria. Now, can I ask you, I'm going to ask you a couple of questions here, everybody. Yeah, Wes, we're going to have a break in about 10 minutes time, and then we're going to come back and finish osmosis. So just bear with us, because I just want to get this chapter done, so then when we come back, then it's just the osmosis chapter, diffusion chapter we have to do. Now, if you look at this here, let me ask a couple of questions here. First question is this. Where's most of the DNA found in our cell? Most of the DNA is found where in our cell? You might help me out here. Quickly as possible. Where's most of the DNA found in a cell? So most of the DNA in a cell is found where? It's found in the nucleus. We know, we'll study the nucleus in a minute. But most of your DNA is in the nucleus. And that DNA, well done, comes from half comes from your father and half comes from your mother. But you need to know that mitochondria also contain DNA. So I want you to note there, apart from the nucleus, mitochondria, and we'll see later, chloroplasts, both contain DNA. Now, does anybody know what they sometimes refer to that DNA as? It's sometimes called non-nuclear DNA. Does anybody know why it's called, this was asked one year, name an example of non-nuclear DNA. Mitochondrial DNA. Do you know what non-nuclear means? It's in the name. Non-nuclear means it's not in the nucleus. So if they ask you, where would you find genetic material, or where would you find DNA outside the nucleus? You'd find it in the mitochondria. Or where would you find non-nuclear DNA? It's the same question, in the mitochondria. Now, the mitochondrial DNA, the DNA in the nucleus we get from both parents. But our mitochondrial DNA, we studied this in human reproduction. Can anybody tell me, who do we get our mitochondrial DNA from? Who do we get our mitochondrial DNA from? Okay? Who do we get our mitochondrial DNA from? Is it our mother, or is it our father, or is it both? So the DNA in the nucleus we get from both parents, half and half. But who do we get our mitochondrial DNA from? Anybody know? Who do we get it? It's from our mother, because we studied this in human reproduction. Now, the reason why we don't get any, and this was a question in your Leaving Cert, in 2016, or was it 15? I think it was 16. Why do we only get mitochondrial DNA from our mother? Because when the head of the sperm, well done everybody, when the head of the sperm enters the egg, there's no mitochondria in the head of the sperm. The mitochondria is found in the collar of the sperm. So when the head of the sperm fuses with the egg, there's no mitochondria from the sperm because they remain in the collar, which doesn't enter the egg. That's in your human reproduction notes. So if you're not too sure about that, you'll see that. And I'll be doing human reproduction soon again for like the 4th time since January. Okay, so note that. That's really important. Now look at this here. You'll see this mitochondria here is inactive. This mitochondria here is active. We know that because look at this here. It's all got to do with, and I'll go over the black here. So look, you see there the membrane. See the membrane? So the inner foldings. Now that mitochondria here that I'm highlighting in black there, you see that there? That mitochondria has very few infoldings. But if you look at the mitochondria down here, I'll go maybe yellow this time. This mitochondria this time has lots of infoldings. Now that increases the surface area, and we studied that in respiration earlier this week. The more infoldings you have, the more that what happens. Does anybody remember what happens in the inner membrane of the mitochondria? Do you remember we studied that on Monday night? What happens in the inner membrane of the mitochondria? It's not the Krebs cycle, but what happens in the inner membrane of the mitochondria? Does anybody remember what happens in the inner membrane of the mitochondria? We know NADH carries electrons and protons to the inner membrane of the mitochondria. But what event happens in the inner membrane of the mitochondria? Has anybody got an answer there? Well done, the electron transport chain. And that's why the more inner foldings you have, my goodness me, that's brilliant everybody. The more inner foldings you have, the more of those electron transport chains can exist and more energy can be produced or released from respiration. So the point I'm making is this. You need to be able to identify an inactive mitochondria and an active mitochondria by looking at the amount of infoldings. So look, if there's very few infoldings in the inner membrane, then it's less energy is released. But if you've got a lot of infoldings, then you're going to have a lot more energy produced. Now, if you're having a really lazy day and you're sitting on the sofa and you say, God, I've done nothing all day and I still feel really tired, it's because your mitochondria look like this. Okay? But then you go for a little walk. And when you go for that little walk, you feel more energized. Because now your mitochondria look like this. And therefore, more inner membrane means more electron transport chains, and therefore more energy can be released. So exercise can increase the number of infoldings in that inner membrane, so more energy is being released. Now, I want to ask, these are all little questions that get asked on this. Where might you find many mitochondria? Well, the question is, why do some tissues need a lot of mitochondria? Because they need more energy, like muscle tissue. We studied that in the last chapter. So many mitochondria are found in muscle tissue or organs like your liver because they require more energy. Less in a plant, many mitochondria are found in the meristem of a plant because in the meristem of a plant, mitosis occurs. And because mitosis occurs there to allow for growth, a lot more energy is needed. So that's why the meristem, which is the shoot tip, the apical bud, the root tip, we studied plants before Easter. We'll do them again in May. So my point is, anywhere you need energy, you're going to have a lot more mitochondria. There's not that many mitochondria found in fat tissue or ground tissue in plants or fat tissue in humans because that tissue doesn't require that much energy. Okay, so before we leave the mitochondria, let's look at the exam questions that can be asked on the mitochondria. Now, the first one, I'm going to actually take you back here. This was a question asked, and it really caught a lot of people out. Do you see this here? Okay, so if you look at this here, name 2 organelles that are found in a typical animal cell that are not visible using a light microscope. Well, if you want to see detail. Now, you may see mitochondria using a light microscope, but you're not going to see them in detail. Okay? So, if you want to see the mitochondria in detail, you got to use an electron microscope. So, you cannot see mitochondria, you cannot see ribosome in detail using a light microscope. Okay? Why is that? Because mitochondria are too small. Ribosomes are too small. Okay, brilliant. If we then go down here a little bit further, what other questions do they ask on mitochondria? You will see here, where are we here? Now, if you look at this, it says, question 24. Apart from the nucleus, name 2 locations in a cell that contain genetic material. Well, genetic material is DNA. So where would you find DNA in a cell apart from the nucleus? The mitochondria, or if it's a plant cell, we haven't studied this yet, the chloroplast. Okay, so state 2 locations of non-nuclear DNA. That's the same question as 24. So mitochondria and chloroplast. Now this one came up last year. See this one here. Place an X and 2 arrows. From the X, place 2 arrows to indicate 2 locations where DNA is found. You could have pointed it to the nucleus because it contains DNA, the chloroplast, which is that there because it contains DNA, and the mitochondria because it contains DNA. There you go. That's been asked like that. Now, if we then go look at question 30, suggest why mitochondria are found in different amounts in different cells. Why do some cells have a lot of mitochondria? Why do some cells have very few? Because they require different amounts of energy. Very easy to answer when you see it like that. Okay, now, if you look at question 33, name 1 type of animal cell that does not have mitochondria. That's not in this chapter, but it's in your blood chapter. Red blood cells don't have a mitochondria. Okay, let's go, and therefore they don't require energy. Now let's go, we've got a couple things left, and then we're done this chapter, and then we're going to have a break. Let's go and look at the nucleus. So the nucleus, of course, okay, the nucleus is found in animal cells, the nucleus is found in a plant cell, okay? Now, if we look at a nucleus, what do we need to know about a nucleus? It's the control center of the cell, that's the first thing. It's the control center of the cell, okay? Now, it's also got a double membrane. You see the double membrane there, the outside membrane and the inside membrane. Now that membrane has pores, meaning that the nucleus is also selectively permeable because of that membrane and the pores it contains. What does your DNA contain? Or what does your nucleus contain? It contains DNA. Now that DNA is found on structures called chromosomes, and you can write that there. The nucleus contains DNA on structures called chromosomes. Chromosomes contain DNA and protein. So we studied that last day. So chromosomes are found in your nucleus and they contain your DNA that's in the nucleus. Now, that nuclear pore allows certain molecules in and out of the nucleus. One such one is mRNA. Now, anybody who was on the platform, we studied DNA and RNA last Sunday and 2 Sundays ago and last Sunday morning to finish it off. And we would have studied during protein synthesis, the mRNA gets the code from the DNA and it leaves the nucleus and it carries that code to the ribosome, telling it what to do. So mRNA is passing out of your nucleus right now to bring a message to the ribosome, telling it what type of protein it should make. So there you go. That's what you need to know about the nucleus. If we quickly go down to the bottom of page 80, okay, the cytoplasm. The cytoplasm is all the living material inside the cell, but outside the nucleus. So the cytoplasm is everything inside the cell except the nucleus. And that includes all the organelles. A lot of people think the cytoplasm is just the liquid part of the cell. No, the liquid part of the cell, the liquid part of the cytoplasm is called the cytosol. But the cytoplasm is all the living material that's outside the nucleus. That includes the mitochondria, the chloroplast, and so on. So that's, and cytosol, as asked once, that was the liquid part of the cytoplasm. Perfect. Other than that then, the vacuole, just note there, plant cells have a large vacuole, you see there? Plant cells have a large vacuole. And we'll study that in the next chapter because it ties in with turgor pressure. Animal cells, the vacuoles are so small we don't really even mention them, okay? So I want you to note there, the vacuole allows the plant cell to store food. It stores sugars and it stores salts. But the vacuole also allows for turgor pressure because when the cell fills with water by osmosis, what that means is this vacuole then, look, when water enters the cell becomes swollen with water. That vacuole then pushes outwards against the cell wall and it gives it strength. That's called turgor pressure. We'll see that in the next chapter. So just note there, the vacuole also gives the plant cell, all right, turgor pressure. Okay, it allows turgor pressure to occur. Okay, finally, the last couple here things, and then we're done here. All right, the chloroplast is the site of photosynthesis. It contains chlorophyll. Now, we did a lot of work on the chloroplast yesterday, so I'm not spending long on this. Just note, though, that the chloroplast also contains DNA. And again, that's non-nuclear DNA because it's not in the nucleus. Perfect. That's what we need to know about the chloroplast. What about the ribosome? The ribosome is the site of protein synthesis. It's where proteins are made. Okay? How does the ribosome make proteins? By joining amino acids together to form a larger complex molecule called a protein. For that reason, protein synthesis is an anabolic reaction. I studied that in the food chapter, but if you want me to write it in here again, I will. It converts smaller molecules. This is why it's an anabolic reaction. Protein synthesis is an anabolic reaction because it converts smaller molecules called amino acids, because they're the basic units of protein, into larger molecules. And the larger molecule is, of course, called a protein. So that's why that is an anabolic reaction. Now, just note, this was asked once. What does a ribosome consist of? Apart from protein, the main component of a ribosome is RNA. All right, RNA. Now you can write the type of RNA called rRNA. We studied that in the DNA and RNA chapter. But there you go. Okay, so the ribosome is composed of rRNA and protein. The last one here you'll see here is the cell wall. Now we know a lot about the cell wall from the food chapter. A structural carbohydrate called cellulose forms the cell wall. What does the cell wall do? It gives strength, but another one is it protects the cell. So you can write that in. It protects the cell or it gives it strength. Okay, brilliant. Let's then go down here. If they ask you to compare the two, a plant cell has a cell wall, an animal cell doesn't. A plant cell has a chloroplast, an animal cell doesn't. Those two should be more than enough. Now let me point this out, everybody. Let me tell you this. If you say a plant cell has a cell wall and you leave it at that, you get 0 marks. You have to say the animal cell doesn't, and then you get your marks. The final thing then, before we have our break, before we come back to the last chapter, a prokaryotic cell, my goodness, put a star beside this one. A prokaryotic cell is a cell that does not have a nucleus. And it also doesn't have membrane enclosed organelles. Now you might say, what's a membrane enclosed organelle? A chloroplast is an organelle that's enclosed with a membrane. A mitochondria, you see it here, is an organelle that's enclosed by a membrane. Now what we're saying is prokaryotic cells don't have them membrane enclosed organelles. So a prokaryotic cell does not have a nucleus, and it does not have membrane enclosed organelles like mitochondria or chloroplast. And our example of that is a bacteria. Look there, there's no nucleus, there's no membrane enclosed organelles like mitochondria. What's a eukaryotic cell? A eukaryotic cell has a nucleus, it's the exact opposite, and it has membrane enclosed organelles. So it has a nucleus, and it has membrane-enclosed organelles like mitochondria and like the chloroplast. And a vacuole, by the way. The vacuole is also a membrane-enclosed organelle. So animal cells, plant cells, for example, are eukaryotic cells. Okay, to finish that off then, let's just take a quick look at our last couple of questions here. If you go to the bottom of page 40, it says here, distinguish between a prokaryotic cell and a eukaryotic cell. A prokaryotic cell does not have a nucleus and it does not have membrane-enclosed organelles. Always give the full definition. You don't know how much they're looking for, so always give the full definition. A eukaryotic cell has a nucleus and it has membrane-enclosed organelles. Okay, brilliant. And other than that, look, it says give two differences between a plant and animal cell. A plant cell has a cell wall, an animal cell doesn't. You gotta say the other one. And a plant cell has a chloroplast, but you gotta say an animal cell doesn't. Okay? Brilliant. So look, you'll go through the few little questions I skipped over, but that's pretty much the main part of that chapter. Okay, now we've done that chapter. I'm gonna give you, it's 6 minutes to 9. I'm gonna give you 6 minutes of a break. And after those 6 minutes, I am then going to ask you to look at, and we're going to spend 35 minutes on diffusion and osmosis. Okay? And I'm going to go through that. It's not as detailed as people think. There's some questions are a bit tricky. So I'm going to give you 5 minutes, everybody. Now, this time, please don't log off. It's half an hour. It's these 7 chapters, remember, that are 30 to 40%. You don't want to know the other 6 and not know osmosis. And then it comes up. My goodness. And you'll be really annoyed. So, I'm going to give you 5 minutes. I'm just going to put the break screen on. It's called, because I need to dip to the toilet and get another glass of water. But I'll be back in 5 minutes, everybody. So don't go anywhere. right, everybody. So this is the last segment of our biology blitz week this week. So we've got osmosis diffusion to look at. Now, we spent 2 hours on all the other chapters. 2 hours on enzymes, 2 hours on respiration, 2 hours on photosynthesis, 2 hours on, what one am I missing? Cell division. All right. So this one, obviously, this class here, a little bit, we're doing a little bit more than just the one chapter because cell diversity and cell structure are so short, but don't take them for granted. That's why I want to still do them, because they do come up. Like last year, cell structure came up and it was 5%. Okay. Could be cell diversity this year, or might be even cell structure again, or maybe both might come up. This sometimes happens. Cell structure could be asked as maybe an experiment. It could be this year. Maybe let's imagine that happened. And that experiment would be 7.5% of your paper. Maybe the plant cell, animal cell, the microscope thrown in there. And then cell diversity comes up as a short question. And all of a sudden, those 2 chapters we've just done in the last hour and a bit could be 12.5% of your paper. If one of them comes up as a short question and the other one comes up as the experiment, maybe. You just never know. And this is the point. They're only easy, and you're only going to do well in them if you know them. And that's why I'm saying... that's why I'm saying... don't be studying, as I say just before we get going here, don't be studying, like just, for example, just don't go and study cell division, enzymes, photosynthesis, respiration, and then leave out cell structure, cell diversity, and osmosis diffusion. Because you'd be really annoyed that 30 to 40%, and you might not have realized that cell structure and cell diversity, osmosis diffusion could have made up 15% of that. You just never know. So we want to make sure we know these 3 smaller chapters. Let's go and look at osmosis diffusion. Now, if you look at this here, I see Dan asked the question before we get going. During cell division, are the chromosomes just floating in the cytoplasm? Yeah, because it depends what stage you're talking about. Dan, sorry, Dan, I had a bit of popcorn there in my break. Stuck in my throat there. But, Dan, in the prophase, the membrane is still around the nucleus. The nuclear membrane is still there, but it's breaking down. So the chromosomes in the prophase are still in the nuclear membrane. But in the metaphase, the nuclear membrane the nuclear membrane is gone. It's broken down completely. So then in the metaphase, the chromosomes then are floating along the equator in a straight line. Okay, so it depends what stage of mitosis you're referring to there. Okay, so let's open up, everybody, on page 106. Again, if you're on the platform today and you're using it and you're not a member, then don't worry. This chapter's notes is there. Okay, I put up these 2 or 3 chapters. Okay, now if you look at this, everybody, and just for the rest, all the other biology blitzes that we're doing and all the other classes that we're also doing, it will be just for premium members. This was, I think, the only week that we're doing for free, just to let you know, just in case anybody was wondering. All right, this here. Last year, 7.5% of your paper. Okay? 2023, 7.5% of your paper. Last year it came up as an experiment. Alright? So, I suppose, let me put it like this. Cell structure came up last year, it was 20 marks. Osmosis diffusion came up last year, it was 30 marks. So, cell structure, the chapter we just done, and osmosis diffusion, the chapter we're doing now, they made up 5 and 7.5. They made up 12.5% of your paper. Those 2 chapters. So my point is, we want to know these easy chapters just as well as we know the more difficult chapters. All right, so we already know. All right, if you want to know what osmosis and diffusion is, you got to have the idea of what a selectively permeable membrane is. And we just did that in the last chapter. It allows some, but not all substances to pass through. It controls what enters and leaves. Now, if you go down here, all right, what components of a cell have membranes and are selectively permeable? Well, we just studied in the previous chapter. The cell membrane is obviously a membrane in a cell. The mitochondria has a membrane around the mitochondria, which is in the cell. The chloroplast in a plant cell, the chloroplast also has its own membrane. The nucleus obviously has its own membrane. So those are the components of a cell that have a membrane. Okay, that was something like that was asked in 2008, and you need to know 2 of them. Okay, now we're going to start off with diffusion. All right, we're going to start off with diffusion. Now, what is diffusion? Diffusion is the movement of molecules from an area of high concentration to an area of low concentration. So it's the movement of molecules, could be a liquid or could be a gas, It's the movement of molecules from an area of higher concentration to an area of lower concentration. Now, because they're going from a higher to a lower concentration, it's passive. Diffusion is passive, which means it does not require energy, which is good news because diffusion is occurring throughout your body right now. Diffusion molecules are going from areas of higher concentration to areas of lower concentration in and out of cells right now in your body, and it doesn't require energy. It's passive. Okay? Joe, does the ribosome also have one? No, the ribosome doesn't have a membrane. So don't use that one as a membrane enclosed organelle. Now, if you then look at this here, everybody, you need to be able to give an example of diffusion in your lungs. Okay? You need to give an example of diffusion in animals. And I always go for diffusion in the lungs because you study this in the breathing system as well. So right now, look at this here, and you need to be able to draw this diagram here. Right now, oxygen, okay? I'll go, what color will I go here? I'll go yellow. So right now, oxygen is going from your lungs, from your alveoli, into your blood by diffusion. So that means oxygen is diffusing from an area of higher concentration in your lungs to an area of lower concentration in your blood, in your capillary. Okay, which gas is going the other way? Carbon dioxide. Carbon dioxide is diffusing. It's going from an area of higher concentration in the blood of the capillary, and it's going, carbon dioxide is moving to an area of lower concentration in your alveoli of your lungs. So there it is. Carbon dioxide diffuses from your blood into your lungs. So that's two examples of diffusion, both occurring in the lungs. Okay, so one more time. Oxygen diffuses from your lungs into your blood. Now, there's so many other examples of diffusion. Right now, we are absorbing nutrients from our intestines into our blood. That's quite often happening as diffusion. So molecules are going from a higher concentration in your intestines, and when you absorb them, they're moving to an area of lower concentration in your blood. That's by diffusion. Now, you also need to know an example of diffusion in a plant. So remember we studied photosynthesis yesterday, and we said carbon dioxide enters the leaf of a plant. Does anybody remember what happens to the carbon that's in carbon dioxide during photosynthesis? Can anybody remind me? Just a bit of a glimpse back to yesterday. What is the fate of the carbon in carbon dioxide in photosynthesis? Now, let me go back to this here while I get that answer. How does carbon dioxide enter the leaf? Through the stomata, by diffusion. The carbon dioxide is going from an area higher concentration in the atmosphere, and it's moving to an area of lower concentration inside the leaf. Okay, oxygen is diffusing the other way. Oxygen diffuses from the leaf out into the atmosphere. Okay, brilliant. Yes, Claudia, you do need to know the definition of a semiplex or selectively permeable membrane. So that's what you need to know about diffusion. Let's go and look at exam questions that are well done. The carbon in carbon dioxide is the carbon that forms glucose. Well done. Now, if you look at this then, everybody, if you look at this here, let me open up osmosis diffusion here. Okay, so all right, here now. Now, if we turn to page 80, just turn to page 80 there. So, first thing is, you need to know the definition of diffusion. It's the movement of molecules from an area of high concentration to an area of low concentration. Brilliant. Now, I have all those definitions in your notes, but they're also at the end of each chapter as well. Now, if you then look at this here, you will then see, let's go down to question 4. Diffusion is passive. Explain the underlying term. It means it doesn't require energy. Okay? Now, name 2 substances that enter your muscles by diffusion. Well, we know our muscle cells require oxygen and glucose for respiration. So how does glucose and oxygen pass from your blood into your cell? By diffusion. All right? Right now, glucose is going from an area of higher concentration in your blood to an area of low concentration in your cell. Right now, oxygen is going from an area of higher concentration in your blood to an area of lower concentration in your cell by diffusion. Okay? Now, look at question 6. It says, in the case of a named molecule, give the precise location at which it diffuses into the body. So what molecule are we going to name? oxygen. Where does it diffuse into your body? It diffuses into your body from your lungs. There you go. You need to know that. You need to know the same for the leaf. In the case of a named molecule, give the precise location at which it diffuses into a plant. Carbon dioxide. And it diffuses into the leaf through the stomata. Okay? Brilliant. Now, if you then go back here, that's what you need to know about diffusion. Okay? Now, if you then go back here, you will then see we're now going to study osmosis. Okay? Now, osmosis came up in last year's Leaving Cert. The experiment came up last year. And they asked this definition. And a lot of people got it wrong. And let me explain why people got it wrong. A lot of people, this is what a lot of people said. I'm going to underline what people said. All right? And now, thankfully, most of my students got it right. But there was one or two, even though I keep saying it to them over and over again, one or two forgot this. Let me show you. Osmosis is the movement of, it's the movement of water from a region of high concentration to a region of low concentration through a selectively permeable membrane or through a semi-permeable membrane. Now, can anybody tell me, and hopefully you notice what I didn't underline, what did those students leave out? They forgot the word water. They said, okay, they said the movement of water. They said it there. But then they said from a region of high concentration to an area of low concentration. That's wrong. A high concentration is the exact opposite to a high water concentration. So you need to use the word water throughout that entire definition. Otherwise, you won't get your marks. So it's the movement of water from an area of high water concentration to an area of low water concentration through a selectively permeable membrane. Okay? Now, we've sorted that. I'm glad we clarified that. Okay? Now, if you then look at this here, Freya just asked, actually, Freya, is the lenticels in the leaf? So the lenticels, the stomata is in the leaf. The lenticels are openings in the stem. They both allow for gas exchange. We study that in plant structure, okay? They both allow gas exchange by diffusion, okay? Now, if you look at this, everybody, let's have a look at what diffusion is. So what we have here, diffusion is the movement of water from a region of high water concentration to a region of lower water concentration across a selectively permeable membrane, which you see there. Now that could be a cell membrane, that could be a membrane that we make called viscine tubing, an artificial membrane. So it's the movement of water from a region of high water concentration to a region of low water concentration. Now that's fine. But sometimes they don't use the word water concentration. Sometimes in the question they might say, all right, it's a low solute concentration. Now what I need you to understand is this. A low solute concentration means it's got not much solute in it. That means the same as a high water concentration. Okay? If it's less concentrated, that means it's a high water concentration. The point I'm trying to make is that's three ways to describe the same thing. High water concentration, low solute concentration, or less concentrated. They mean the same thing. Okay? Because if it's less concentrated, it means it's got less solute in it, which means it's got a higher water concentration. Really important that. That's so important in terms of answering questions that. Now look at the other one. If it's more concentrated, that means it's got more solute in it, more salt or sugar, for example, and therefore it's got a lower water concentration. So you can clearly see that the solution in the left here, the solution on the left has a higher water concentration, but the solution on the right has a lower water concentration because the solution on the right has also got solute in it, which means it's more concentrated. Okay? Really important that you know that those three terms reference the same thing. All right? Now, here is a question here. Look, how does the solution B differ from solution A? Solution B is more concentrated. That's how you describe that. Or solution A is less concentrated. Okay? So solution B is more concentrated. Or you could say got more solute in it. Okay, that's how you answer that question, and that's been asked something like that in your Leaving Cert. Now, if you then look at this here, this hasn't come up in a long time. It came up twice, but it hasn't come up in a long time. Okay, it says, why is osmosis a special type of diffusion? You're basically given the definition here, because it's the movement of water from a high water concentration to a low water concentration across a semi or selectively permeable membrane. Osmosis is the same as diffusion in that it's also passive. It doesn't require energy. So the difference between osmosis and diffusion is the movement of molecules from a high to a low concentration, and it's passive. But osmosis is only the movement of water from a high water to a low water concentration across a selectively permeable membrane. So diffusion doesn't require a membrane. All right, diffusion can still occur through a membrane, but it doesn't need a membrane for it to occur. Osmosis does. Okay, so that's why it's so important we know that. Now, you need to then know how osmosis is related to food preservation. So if we put a load of sugar in jam, or if you salt meat, obviously we don't really salt meat for now for preservation much anymore because we have fridges and freezers. But if you go back a couple hundred years ago, that's how they would have preserved the meat, even less. Okay, they would have salted the meat. Now you need to know, how does salting meat preserve the meat? So what and I've summarized it here in one sentence, but I'm just going to show you here in a diagram. So can you imagine, here is a piece of meat. Imagine that's your piece of meat. And we salted the meat. So on this piece of meat, there's bacteria. And the bacteria is going to try to make the meat go off so it's not edible. So we want to make sure, we want to kill the bacteria so it doesn't cause the meat to go off. So what you do is, you add a load of salt to the meat. Okay? Now, when you add a load of salt to the meat, look what's happening. When you're adding a load of salt to the meat, okay? When you're adding a load of salt to the meat, hold on now, here we go. So when you're adding a load of salt to the meat now has got more solute in it. So therefore, the meat now has a lower water concentration. So now, the outside the meat has the meat has a lower water concentration than the bacteria. So the bacteria has the higher water concentration inside, and the meat has the lower water concentration because you've salted the meat. So what way does the water move? Water always goes from a higher water concentration, in this case inside the bacterial cell, to an area of lower water concentration, in this case the meat, because you added a little salt to the meat. That's why it's created this environment. So the bacteria, let's highlight this now, the bacteria lose water due to osmosis and they die. And because the bacteria die, the meat then is preserved. Okay, it's going to be preserved for much longer. Now I've seen that being worth 9 marks. That there was worth 2.5% of your Leaving Cert 1 year. Okay, now it's unlikely to be asked that much again. It's probably more likely to be 6 marks. But bacteria lose water and die due to osmosis. When you salt meat, the bacteria can't live on the meat because you create an environment that has a lower water concentration on the meat and a higher water concentration then therefore in the bacterial cell. And the water then moves out of the bacterial cell due to osmosis and dies. Okay, that's been asked in your Leaving Cert. Now, we're going to look through this then, right? We're going to look through this here. Osmosis and animal cells. Now, this is quite straightforward if you understand the basics, and hopefully now you do. Water goes from an area of high water concentration to an area of lower water concentration across a selectively permeable membrane. Now, if you look at this here, you've got the same water concentration inside and outside. Okay? So, if the animal cell is in a solution that's got the same concentration as the cytoplasm. If that's the case, if the same water concentration is inside the cell as outside the cell, water will move in and out at the same rate. Okay, so just note there, that allows water to enter and leave the cell at the same rate due to osmosis. Alright, and that's how animals live in seawater, because their cytoplasm has the same salt concentration, the same solute concentration as the seawater. So then they don't have to try to deal with the issue of living in salt water. Okay, so that's how they survive in salt water. The environment inside the cell has the same salt concentration as outside in the water, and therefore water will enter and leave at the same rate. So the cell won't burst or it won't shrivel up. Okay, that's in that one. Now look at this one here. if you put an animal cell in a less concentrated solution. So look here. The best way to visualize this is this. Here is an animal cell. Now that animal cell is in an environment, look up here, that's less concentrated. Now less concentrated means what? Less concentrated means it's got a higher water concentration. You see that there? So if it's less concentrated, it's got a higher water concentration. So I'm going to go here. So the animal cell is in a less concentrated external solution. So the external solution is less concentrated. So less concentrated, which means higher water concentration. So outside the cell, so here's this animal cell here. Outside the animal cell has a higher water concentration because we just said, for that reason, it's less concentrated outside the cell, so it's got a higher water concentration. So that means inside the cell has the lower water concentration. For that reason, due to osmosis, water always goes from an area of higher water concentration outside to an area of lower water concentration inside the cell. So that animal cell now has a problem. That animal cell is going to burst. So look here. If an animal cell is in a less concentrated solution, what does that mean? And this is why I'm always saying, go back and look at this. Less concentrated means higher water. So if an animal cell is in a less concentrated solution in itself, it's in a higher water concentration than compared to inside the animal cell, water due to osmosis will go from a higher water concentration to an area of lower water concentration. So the animal cell gains water due to osmosis. It will enlarge, burst, and die. Why does it burst? Because it doesn't have a cell wall. Okay, so this was a question one year. What do you think would happen to an animal cell in a less concentrated solution? Less concentrated means higher water concentration. That means water enters it and it bursts. Okay, now let's look at the opposite scenario. Let's go to the one on the next page, page 110. Okay, I'm going to skip the amoeba for now because we study the amoeba in another chapter. But if I have time, I'll reference it as well. But let's have a look here. If an animal cell is in a more concentrated solution, that's the exact opposite. So look at this here now. If the animal cell is in a more concentrated solution, so let me go back and show you this here. If it's more concentrated, it means it's got a lower water concentration on the outside. So if the animal cell, you see here now, if the animal cell is in a more concentrated external solution, so the outside environment is more concentrated, that means if it's more concentrated, it's got a lower water concentration. And due to osmosis, water always goes from an area of higher water concentration to an area of lower water concentration. Inside the cell has the higher water concentration. Outside the cell has the lower water concentration because it's more concentrated, more solute in it. So therefore it's a lower water concentration. So due to osmosis, the animal cell will lose water due to osmosis. The animal cell will shrivel and it dies. And you don't want that to happen. So look at this here. This is a red blood cell that's gaining too much water. Due to osmosis, it bursts and dies. This here is a red blood cell that's losing water due to osmosis, and it shrivels up and dies. We don't want either to happen. We want our red blood cells to have the same amount of water entering and leaving the cell by osmosis. And that's why our kidneys are so important, because our kidneys controls the salt and the water concentration of our blood. So our liquid part of the blood has the same concentration as the cells. So therefore water will enter and leave the red blood cells at the appropriate rate. Okay, so look at this here. State one location where osmosis occurs in humans. Water moving from your large intestine into your blood. Water right now is being absorbed from your intestines into your blood by osmosis. Okay, now let's look at a plant cell. It's the same thing applies. Admin, you might give me 10 extra minutes and we'll finish this chapter. It'll only take 10 minutes, everybody. So let's look at osmosis in plant cells. Now, osmosis in plant cells, same idea. We're sticking to the same environments, right? So what happens if a plant cell is in a less concentrated external solution? So there's the plant cell. So the outside environment has a less concentrated solution. Now, what does that mean? That means, let's go back here. That means if the outside environment has a less concentrated solution, that means the outside environment has a higher water concentration than the inside environment. So let's go back and just write that there, because it's all about visualizing this. So the outside environment has a less concentrated solution. We said that there, which means it's got a higher water concentration. That means the same thing. So if outside the cell has a higher water concentration, that means inside the cell has a lower water concentration. So therefore, water enters the cell due to osmosis. Now, if that was an animal cell, it would burst and die. Plant cells don't, because they have a cell wall. So water moves into the cell by osmosis, but it doesn't burst. Why? Because the cell wall prevents it from bursting. Not only that, it also allows turgor pressure to exist. So we're going to tie this now in with turgor pressure. Okay? Now, let me show you this here. Okay? Let me show you this here. When water, get me a pen here. Now, when water enters the plant cell, see water entering the plant cell. Water enters the plant cell. Okay? The vacuole now becomes swollen with water. See the vacuole there? That vacuole becomes swollen with water. What that does then is, that vacuole then, because it's swollen with water, because water has entered the plant cell by osmosis, that vacuole then pushes out against the cell wall. So the black arrows represents water moving into the cell by osmosis. And then the vacuole becomes swollen with water. That then creates an outward pressure, because now the vacuole is pushing out against the cell wall because it's much bigger. And that gives the plant cell strength. Because without the vacuole pushing out against the cell wall, the plant cell would shrivel up and die. So I want to note there, turgor pressure, what is it? It's the pressure of the cytoplasm in the vacuole. So there's the vacuole there. See the vacuole there, everybody? So the vacuole becomes swollen with water when water enters the cell by osmosis. And that then results in the vacuole pushing out against the cell wall. So I want you to just note there, that means it's then pushing out against the cell wall. That's what turgor pressure is. One more time. It's the pressure of the vacuole pushing out against the cell wall. Now, what condition do we describe this as? The condition is turgid. Be very careful. If they ask you, all right, this plant cell, what condition is this showing? It's turgid, not turgor pressure. Turgor pressure allows the cell to become turgid, but the condition is turgid. That's very strict on that a few years ago. Okay? Now, if you look at this, everybody, why is turgor pressure so important? It gives the cell strength and support. Now, if you look at this, you will see this cell on the left is a turgid cell. Why? Because water is entering the cell by osmosis. And because water is entering the cell by osmosis, then, I'll get a different color here, then that vacuole then pushes out against the cell wall. So the water moving in results in the vacuole becoming swollen with water, which means then it pushes out against the cell wall. So that there means the vacuole pushes out against the cell wall, and the plant cell is turgid for that reason. The cell over here, see this cell here? This cell has lost turgor pressure. Why? Because water has left the cell by osmosis. That means the vacuole is shriveled up. See that there? So that vacuole is not pushing out against the cell wall, because water is leaving the cell. The vacuole now is shriveled up. So now that plant cell shrivels and it dies. Okay? So I want you to note there, put a star beside this, the bottom page. A plant cell remains turgid if they take in as much water as they lose. Once they start losing more water than they gain, then the vacuole starts to shrivel, and therefore then it doesn't push out against the cell wall anymore. And then the cell begins to lose turgor pressure. Okay? Look at this here. What two features allow a plant cell to become turgid? The cell wall, because that means when that outward pressure exists, the cell wall prevents it from bursting. And what creates the outward pressure? It's the vacuole. All right? So that's why turgor pressure occurs. The cell wall prevents it from bursting, and the vacuole is the part that creates the outward pressure when it becomes swollen with water. Okay? Brilliant. Now, if you then go down here, thanks, admin. 10 minutes should be is all I need there. Thank you so much. Now, if you then look at this here, okay, what happens if plant cells are in a more concentrated external solution. We did the same for animals. Let's go through it for plants. Look here. If the solution is more concentrated, that means it's got a lower water concentration. So in this scenario here, all right, if plant cells are in a more concentrated solution, so outside the cell is more concentrated, which means it's got a lower water concentration. So outside the cell has a lower water concentration. That means, due to osmosis, water leaves that cell. And then, for that reason, look at the vacuole. That vacuole is shriveled up. That vacuole is not pushing out against the cell wall anymore. And that plant cell has lost turgor pressure. Now, what do we say happens to the plant? The plant wilts. You have to use that word to describe a plant. If you say that the plant has lost turgor pressure, no, 0 marks. The plant wilts. Now, the cells of that plant, you see that plant there? That plant's cells look like that. Because the cell membrane and the vacuole, you see there, see the gap there? See that gap? That gap shouldn't be there. That cell membrane should be pushed out against the cell wall if the vacuole and the cell had enough water in it. But obviously this plant cell doesn't, because it's losing water instead of gaining water by osmosis. And for that reason, the plant wilts. Really important we use that word. The plant wilts. Look at this here. How can a plant cell return to become turgid again? Well, you place water, you add distilled water, you add water to the soil. Now, and then therefore water enters the plant by osmosis through the root hairs, and then the water then enters the cells through the plant by osmosis. Look at this here. Name the substance that causes the change in mass in plant tissue. It's water entering and leaving by osmosis. When water enters, it gains mass. When water leaves, it loses mass. That was a Leaving Cert question. Now, I'm not going to do the experiment today for 2 reasons. Number 1, because I don't have time. I'm about out of time, right? I'm going to do 7 minutes of exam questions. The other reason, because the experiment did come up last year, and you had to describe the procedure. Like all the experiments over the last 2 years, you had to describe the procedure. Just because I'm not doing it doesn't mean it won't come up again. Like, it doesn't happen very often. Experiments come up 2 years in a row. Now, this year, you might have an experiment question 9. This is question 9, the osmosis experiment. Comes up as part of question 9 with 7 other experiments, or 6 other experiments, and this is the number 7. It might be this experiment with 2 other experiments. So this one could come up maybe partially, or maybe this question was answered poorly and they want to put it in again. You just never know. So just make sure you look over it. Okay? Now, I'm going to do a couple exam questions now on what we did about plant and animal cells and osmosis. So if you look at this here, all right, if you look at this here, you will see, let me show you this here now. Here we go. Now, if we look here, let's see here now. Question 15. Explain the importance of turgor pressure for plants. It gives the plants strength and support. Question 17. Give 2 features of a plant cell that allows it to remain turgid. It's got a cell wall and a vacuole. Now, if you then look at this here, okay. Question 19. Can animal cells become turgid? No. Why? Because they don't have a cell wall. They would burst because they don't have a cell wall. All right. Now, if you then look at this here, question 21. What happens to a plant What happens to a plant if its cells lose turgidity? If you say shrivels, you probably won't get your marks. You got to say wilts. That's the word you use in the marking scheme. It wilts. Okay. Now, if you then go down here a little bit further, all right. Now, I don't have time to do all these questions, and there's a lovely question there. Okay. There's a lovely question here. I'll spend a couple of moments looking at it. So you will see here, you got question 24. You got different pieces of potato in different solutions, different sucrose solutions. So look, there's A, B, C, D, E, and F. A, B, C, D, E, and F. Now, in those 6 beakers, you have got a piece of potato. Okay? A piece of potato. Now, what this graph is showing you, anything above 0 means it's increased in mass compared to the control. This is the control here, okay? Now, anything below 0, it lost mass. So you can see A and B gained in mass. That means water entered these cells by osmosis because it gained mass. So water entered these cells by osmosis because of the solution that it was in. Now you will see D, E, and F, these three lost mass. Why? Because water left those potato cells by osmosis, so it lost mass. You will notice this one's in C. This one didn't change mass at all. See this here? Didn't change mass at all. Why? Because water entered and left at the same rate. Okay? Now, because we know that, it says determine the concentration of the contents of the potato cell. So which one of these do you use to determine the concentration of the potato cells themselves? Well, you look at the one that didn't change in mass. Why did it not change in mass? Because the potato cells had the same solution, same concentration as the solution that it was in. Because water entered and left that potato cell C at the same rate. So you go to C, you bring a dotted line down, and that indicates the concentration of the potato cell. Because at that concentration, those potato cells did not lose any water because it had the same concentration as the solution that it was in. So you can see there, they accepted anything from 0.3 to 0.35. Admin, you might give me 5 more minutes. I need 5 more just to say my goodbyes. Now look at this here. Look at part B. Explain in detail why pieces of solutions in D and to F lost mass. Well, they lost mass because, look, water left those potato cells by osmosis. They lost water by osmosis. All right, now that got you full marks, but if you want to got the same, why? Because the sucrose solution was more concentrated. Because it was more concentrated, it had a lower water concentration because the solution was more concentrated. So therefore, water moved out into the solution. Okay, perfect. Now, it says then, give one example, an example of this mechanism described in B for food preservation. Salting meat, sugar and jam. It's based on the same principle. Now, if you go down here, okay, I want to do this then, and then I'm going to finish off with one last little thing. This came up a few years ago, and a lot of people didn't like it. About 4 or 5 years ago, this one, right? So you're looking at plant cells, okay? And these plant cells, A and B, are placed in 2 different solutions. Now, you can clearly see the plant cells in B have lost turgor pressure because look at the gap between the cell wall and the cell membrane. See that gap there? All right, because it's shriveled. All right, the vacuole is not pushing up against the cell wall. So it says, name the condition which describes the cells in A. Well, they're turgid, not turgor pressure, turgid. Turgor pressure got you 0 marks. Describe how these cells maintain the shape. Turgor pressure, or explain it. It's the pressure of the cytoplasm and the vacuole against the cell wall, or you could say turgor pressure. Now, question C. How did the solution B differ from solution A? Well, because water left these plant cells, that meant that the solution that they were in had a lower water concentration. Okay? And if it's got a lower water concentration, the solution that these plant cells are in, that means low water concentration means the solution has got more solute in it. So there's your answer. Solution B has more solute in it than A. And that's why water went from a higher water concentration to a lower water concentration out of the plant cell, and therefore they lost turgor pressure. Name and describe in detail the process which has occurred to the cells in B. The cells in B have lost water due to osmosis. So that's the name of the process, and you had to describe it. The movement of water from a high water to a low water concentration across a selectively permeable membrane. Look at question E. How could these cells in B be restored? To A, how could you get these cells to look like these cells? You could place them in water again. Okay, brilliant. Now, there is more questions on that, but I'm running out of time, and I want to say one last thing. I'm not doing that experiment with you today because I just don't have time, and it came up last year. But I do want to say this to you. Active transport is the last thing on this page, or on page 114. I'm not going to spend long about it. Admin, did you give me a few extra minutes there? You might just confirm for me one last time. So, active transport. It's the opposite to diffusion and osmosis. Diffusion and osmosis is passive. They don't require energy because they're going from high to low. Active transport is the opposite. The molecules are going from against a gradient. It's like running up a hill. They're going from a low concentration to a high concentration. So they're going from a lower to a higher concentration. Because they're going from a lower concentration to a higher concentration, that requires energy. So it's the opposite to passive. It is active, which means it requires energy. It requires energy. All right. Now, where does active transport occur in plants? Well, the root hairs of a plant absorb minerals. Root hairs absorb water by osmosis, but root hairs absorb minerals from the soil by active transport, which means the minerals are going from a lower concentration in the soil to a higher concentration in the root. And that's why root hairs have a large number of mitochondria, because root hairs need a lot more energy because they need to absorb minerals by active transport. You will also see active transport occurs in humans. In the nephron of the kidney, all right, the proximal convoluted tubule of the nephron of the kidney, it reabsorbs glucose and amino acids by active transport, which means molecules are going from a lower concentration to a higher concentration. Now, I'm not going to go into that today because we study the kidney in a whole chapter, but that's where active transport occurs there also. Now, look, that last chapter, I would love to spend another half hour on it, but I would love to know a few more questions on it. But you have all the questions there. All you have to do is practice them. So what I want to say to you is this, because I know there's a lot of people here attending free, and then some people are just here, obviously premium members, they're here as well. Let me just remind you, all right? For the free members, for the free people here for the week, I hope you enjoyed it. I know the chat has been switched off for you there today and yesterday because it was just too crazy. There was 300 or 400 people on, and it was just too much going on. So we said we just keep it so you could just focus on the class instead and listen instead. So for premium members now, look at this. All right, your next, I'm doing Sunday. This Sunday is normal. Okay, 6 to 8. Tuesday normal, and Thursday normal. Next week's normal, back to normal. Then the week after that, then again, normal. But then, look at this week. We're going to do and I say, what I mean by normal, Sunday, Tuesday, and Thursday. But then, May the 10th, we're going to do the 5 big human biology chapters. Okay? Now, the week before that, in the Sunday, Tuesday, and Thursday class, I'm going to be doing some other human biology, the breathing system. So the week before this Blitz, I'm going to be doing some of the smaller human biology. And then I'm going to be doing the 5 big human biology here. So the week before the 10th of May, I'm going to be focusing the Sunday, Tuesday, and Thursday class on small human biology chapters. And then we'll do the week after, which is this week here, on the 5 big ones. So those 2 weeks will get most of human biology done. Okay? And then, everybody, look here. Then the 12th, 24th, the 28th, I'm going to be doing the 3 experiment questions, 8, 9, and 10. I'll do the experiments in each one of those. All right, the 8 experiments, the 7 experiments, and the 7. And then Unit 1, the last 2 days as well. But let me tell you this. We have so much more planned for so many subjects. I'm going to Cork on May the 10th to do DNA and genetic crosses in the Clayton Hotel in Cork City. May the 25th and 26th, I'm doing 2 days in the Talbot Hotel. But them same 2 days, in the evening time, I'm doing question 9 and 10. So May the 25th and May the 26th, I'm doing 2 hours in the morning, basically, and 2 hours in the evening. All right, the ones in the morning are in person, the ones in the evening are online. So you can pick and choose whichever ones you want to come. I don't expect you to be at everything. But the point I'm trying to say is there's a lot of classes coming at you if you need them. And you'll attend the ones you need. And don't forget this, of course, everybody. Don't forget the on-demand. All right, you'll see all the on-demand stuff there. Look, all the videos on all the chapters. You'll see there. Video on ecosystem, and then the same video on exam questions based on that content. And I do that for the all. And I know genetic crosses and plant reproduction are on the on-demand and have not been done out fully, but don't worry, you still have them there, the live classes. Oh, jeepers, where did that go? The live classes there. You'll see genetic cross 1 and genetic cross 2, and then you go over here, look, and then you'll see the two classes on the Thursday one, sorry. You see the Thursday one here, where we did plants. So look, there's the 2 chapters on plant reproduction. So you have all that there. So all of it's on demand, and those 2 chapters you can view there as well. Look, everybody. If you're premium members, brilliant. Great to have you again. If you're free members and you came for the week, I hope you find it useful. I have to say the numbers were just unbelievable. I think there was nearly 350 here tonight again. So well done. Keep working hard. And I'll say this to you to finish off. It's all about sections, everybody. It's all about sections. Study Unit 1, 25 to 32.5%. Study them 7 chapters in Unit 2, 30 to 40%. Study these 4 chapters in Unit 3, should be 10 to 20%. It won't be that far away. Study your human biology, 30 to 45%. You get the idea. All right? And yes, people keep asking me, if I leave out genetics, can I still do a full paper? You can. And you'll still get a H1. If you're looking for that H1, you'll just have less options. Maybe still do the DNA experiment though, because it's part of the first 8 experiments. Or maybe you study genetics and you want to leave out plants, the 5 plant chapters. There's going to be probably a good few questions on plants this year, because there wasn't that many last year. But even so, you'll still be able to do a full paper. So you could leave out plants if you had to. Ideally though, is try to study some of plants. Don't rule out all of it. Plant structure, plant transport, they're lovely chapters. Go to my on demand, you'll see it. All right, but I'll be doing most of the course again over the next, so from this week onwards to the end of the year, I'll have pretty much every chapter revised again. Okay, doing live. Okay, thank you so much, everybody. Anybody got questions there? Brendan Freya, I'm so delighted you found the week useful. Are you planning on doing any more live classes on plant chapters? Yes, Sophie, you must have been listening, Sophie. This Thursday, or this day next week, I'm going to finish off plants because we've been doing plants over 4 Thursdays or 5. So I'm going to spend next Thursday on it. But I'm also doing plants in a few master classes in April or in May as well. So you'll see the timetable. There's a whole timetable coming out for the month of May. And I'm telling you, when you see the biology, you'll be blown away because we're doing 2 biology blitz. There's another week that I'm doing another 5 classes in a row. There's some days I'm doing classes twice a day in the morning and the evening. So you can pick and choose. It'll all be there and you can see what I'm covering each one. All right. And the weekend before you're leaving cert and the night before you're leaving cert, I'll be there to do Q&A sessions. If anybody wants to come on and ask me a question, I'll be there just sitting there and I'll be covering a few things as well. All right, everybody. I'm fairly confident now on Unit 1 and 2. How should I navigate Unit 3? Well, Michael, maybe start off with the 4 chapters at the start. 10 to 20%. You'll get them done in no time. And that's another little, hopefully, it should be 10 to 20%. I don't think it'll be much less. A couple of years ago it was 22%. But, and then that's another little bit done. And then you can start, and then Michael, come to my biology blitz for the 5 human biology chapters, the big ones, and that'll break the back of that. And you'll say, God, I feel pretty good now about human biology. So that's what I would do. Start with those 4, and then make sure you attend my biology blitz for the 5 human biology chapters. All right, everybody, I really appreciate the thank yous, and I'm so glad you find it so useful. Which topics are we doing next week on Tuesday and Sunday? You must have been, Sophia, you must have been a bit late there coming in today. Maybe you weren't, but there you go. Look, there you are, Sophia. Sunday, variation and evolution and genetic engineering. That's the end of the genetic section because I've done all the other genetic chapters over the last few Sundays. Tuesday, I've never done the lymphatic system. It's a really short one, and I'm going to go back and do the senses again because I did them about 3 months ago. And then I'm going to spend Thursday finishing off plant biology, and that's plant biology then done. All right. I'm delighted, everybody, that you find it useful. Join the mailing list for masterclasses, everyone, so you don't miss out. Oh, there, Kitty. Well, good advice. But Kitty and everyone, the timetable is coming up for the next month, and I advise you very strongly to look at it. There's so many subjects doing so many extra classes, and I'll be doing just more and more stuff like this. It's all about the more you understand it, the less learning you have to do because you know it, you understand it. So obviously you still have to learn the definitions, but if you feels that you understand this better, it means you're going to spend less time studying it because you understand it. That's how it works. All right, everybody. Thank you so much. Have a lovely day. And I really appreciate all the thank yous. I don't think I'm missing any questions here. Oh, my live chat has frozen here. I can't go back down, but I think we're all good. Oh, there we are again. Let me see here. Any other? Sive. Hypertonic, hypotonic, isotonic, they're not on the course. Those terms are not on the course. Okay? So those terms are not on the course, all right? Just in case you're wondering. Any more live classes on plants? I think I answered that. Any other questions? I think we're all good. I think we're all good. Chris says, could you say turgor pressure is the force within the cell that pushes the plasma membrane against the wall? I don't know, Chris. I think you're nearly there. I would emphasize it's the cell, it's the vacuole and the cytoplasm pushing out against the cell wall. It pushes the cell membrane out against the cell wall. I would just throw in the vacuole there as well and pushing out against the cell wall. All right, everybody. Thank you so much. I'm going to say good night. See you all later. I'll see you Sunday, 6 o'clock. Good luck.